Yeah, thank you for the kind introduction. And what I'm trying to do in the next 30 minutes or so is to review the evidence of supporting the implementation of precision medicine in cancer care. I guess we now know what we can do. So what is the clinical evidence? So the clinical utility. So this is the outline of my talk, and I'm going to start with some definition, so back to the basics. So what is precision medicine? So according to the National Cancer Institute definition, it's a form of medicine that uses the information about a person's genes, proteins, or environment to prevent, diagnose, or treat disease. So in the field of oncology, it is to prevent, diagnose, or treat cancer. So in fact, precision medicine has been a reality in oncology for years now, as you will see. We are looking for BRCA1 mutations to prevent breast cancer or ovarian cancer, as an example. We assess the FLI1 EWS translocation to diagnose Ewing sarcoma. We are looking for EGFR mutations or ALK translocation to seek for treatments to treat non-small cell lung cancer when, with the matching drugs. In terms of proteins, as an example, estrogen receptors are very important either to diagnose breast cancer or to treat breast cancer. And environments or family history, as an example, might help preventing breast cancer. Asbestos might help diagnose mesothelioma, and the ECOG performance status might help deciding what treatment to give to cancer patients. So precision medicine is a reality, and we all know it now. So the question is, do we need high throughput technologies for cancer care today? And there are 2 points. So we might need high throughput technologies either to prognose or to have a predictive value, so to predict response to treatment. So the prognostic value of high throughput technologies is already done with the gene signatures in patients with early breast cancer who are mainly treated with surgery and radiation therapy. But there are gene signatures such as MAMAprint or Oncotype DX that will tell you what is the prognosis of these patients and whether we need to give them chemotherapy or not. So high throughput technologies assessing RNA in that case is already validated in patients with early breast cancer. Now, what is the predictive value of high throughput technologies? In other terms, and especially in patients with recurrent cancer where surgery and radiation therapy no longer are useful strategies except for palliative care. So do we need high throughput technologies to guide our treatments and knowing what drugs to give? And that's the question I will try to answer now. And I want to review the evidence of that with you. And there are several types of studies that have been performed to try to answer that question. First one are the meta-analysis. Meta-analysis of phase 1 trials demonstrated that the response rate to patients included in a phase 1 clinical trial based on a molecular alteration was much higher than the response rate in patients included in phase 1 trial without any molecular selection. And this was the same for PFS, actually. Now, if you look at this study, and only for drugs that are approved, it is true. Now, if you look at the subgroup of drugs that are not FDA approved or EMEA approved, there is no longer any benefit here. And this is the same here for the PFS. So here we know that having a molecular selection to drive therapy is validated, and this is validated for drugs that are already on the market, such as Herceptin, erlotinib, etc. But for this kind of drugs, obviously, you don't need to use high throughput technologies, but only a single gene assay is fine, right? In this meta-analysis for phase 2 trials, we had similar results in terms of response rate, which was much higher in patients who got personalized treatment, and this was the same for PFS and OS. Now again, if you look at the subgroup of the drugs that are not approved today, there is no longer any difference between the not personalized approach and the personalized approach. So from this meta-analysis, there is no robust data suggesting that we should use high throughput technologies, right? So now let's go to the retrospective studies, and one of the biggest study has been the impact study by the MD Anderson Cancer Centers, where they used for any phase 1 for any patient referred for a phase 1 clinical trial at the center to have a molecular profiling on their tumor, and then to try to send them, to guide them to phase 1 clinical trial with the match therapy based on the identified molecular alteration. And what you can see here is that retrospectively, patients who did receive match-targeted therapy had a response rate of 12%, as compared to only 5% in patients who were sent to phase 1 clinical trial without any match-targeted therapy. And this translated into an improved progression-free survival and an improved overall survival as well. Now, the drawback of this kind of retrospective studies is the retrospective aspect of the study. And I explain this to you with this example here. This is an example of PFS curves of patients with lung cancer treated with cisplatin. Here is the curve of patients who were treated with cisplatin and had an EGFR mutation, and the red curve is a group of patients who were treated with cisplatin-based chemotherapy and who had no EGFR mutation. And you can see that there is a difference between the two curves. Now, obviously, the difference is not related to the fact that cisplatin only works if there is an EGFR mutation, right? It has no relation between the EGFR mutation and the cisplatin chemotherapy. But it's only the fact that non-small cell lung cancer patients who have an EGFR mutation have intrinsically a better prognosis than patients who do not have any EGFR mutation. So the bias of retrospective studies is that it cannot distinguish between the prognostic value of the molecular alteration and the predictive value. And what is being shown here for single molecular alteration holds true for a complete molecular profile as well. Maybe patients who have a molecular, druggable molecular alteration have a better prognosis than patients who do not have. That we don't know. So that's why we cannot draw any robust conclusions from this retrospective analysis. So now we are lucky, we have some prospective studies. And let's start with the prospective non-randomized trials. And really the first study has been the one by Von Hoff and colleagues published in the JCO in 2010. And what they did is that patients with any kind of cancer had a tumor biopsy taken, and they used to analyze the biopsy and looking for gene expression abnormalities and doing also immunohistochemistry and FISH. It was back in before 2010, so almost 10 years ago now. And depending on that, they decided to treat or not a patient with a match therapy. Therapies could be targeted agents, but also hormone therapies, or even chemotherapies, or sometimes drugs given outside the field of oncology, such as metformin. And the way they did to assess the efficiency of that strategy was to look at the progression-free survival on the last treatment that the patient received, which was a non-matched treatment, obviously, and the treatment that the patient received based on the molecular profile. And they evaluated the proportion of patients who had a PFS ratio exceeding 1.3. And these are the results. They had 27% of patients with a PFS ratio exceeding 1.3. So as an example, that last patient here had a PFS of 2 months on the last treatment he received, and a PFS of 14 months on the treatment he did receive based on a molecular alteration that was identified using the molecular profiling. The Gustave Roussy did a similar study, which was the Moscato study. And in fact, they got similar results. They had also 33% of patients with a PFS ratio exceeding 1.3. So suggesting that this approach to molecularly profile cancer patients and then to give them matched therapy might be beneficial for 27 or 33% of patients. Now, what is the issue with this kind of studies? So first is the timing of assessment. Obviously, if you think that patients before on PFS1 have a CT scan every 6 weeks, right? And then patients on matched treatment have a CT scan every 8 weeks, then you will get 100% of patients with a PFS ratio exceeding 1.3, just because of the timing of the assessments. I do not mean with that this was done in these studies, right? But the timing of the evaluation, of the tumor evaluation, is very important. Second point is the method for the assessment of the efficacy. So usually patients, at least in the Moscato trial, were sent to phase 1 clinical trials, and we believe that these patients were assessed using the RECIST criteria. Now we don't know what happened on the last treatment. Were these patients assessed according to RECIST, or a patient who had a clinical progression was considered as having a progression, which in that case is different from RECIST, right? And again, if the method of evaluation is not the same on both treatments, then the conclusions cannot be robust. And that's why these papers are not level 1 evidence of precision medicine in this subgroup of patients. So now we move to prospective randomized trials. And unfortunately, we only have one trial we did perform at the Institut Curie and in other centers in France. And what we did actually was to include patients with any kind of cancer that was refractory to a standard of care, perform a tumor biopsy and sequencing using a targeted sequencing and a cytoscan HD for gene copy number alterations, and also immunohistochemistry for hormone receptor status, and also to confirm or not any amplification or deletion that was detected using cytoscan HD. All bioinformatics was done at the Institut Curie, and the molecular biology board used to meet on a weekly basis to discuss the case. And if a druggable molecular alteration was identified, then patient would be randomized between the matched targeted therapy or what we would have done in the clinic. So I know colleagues from the US told me, so your control arm should be no treatment, right? Because all patients already got standard of care treatment. But I mean, in France and in many other countries, I guess in Europe, even when a patient has failed standard of care therapy, if the patient is doing well, we will still try something else, such as a capecitabine in colorectal cancer or mitomycin C or I don't know. What we allowed is that patient could cross over disease progression, so meaning that if they were randomized into that arm, they could cross over to targeted therapy and reverse way, actually. So this is the treatment algorithm we used for this trial. So maybe looking at this treatment algorithm, you would say, oh, what did they do? They did give like everolimus for a patient with a PI3K mutation. This doesn't make sense. This treatment algorithm was set up like in 2011, so it was like 7 years ago now. So I agree, the literature now is ongoing, and probably you would have done differently today. What I know is that all ongoing precision medicine trials, they have treatment algorithms that are not much more complicated than this simplistic one, actually. Although I know it's probably one of the limitations. So for patients with hormone receptor expression, they did receive hormone therapy. For patients with mutation or amplification in an oncogene such as BRAF or EGFR, etc., they did receive the match-targeted therapies. And for patients with alteration on the PI3K AKT-mTOR pathway, all patients did receive everolimus because these were the drugs that were available in the frame of the clinical trial at that time. As you can note, all patients received only a single agent therapy, except for patients who had an HER2 alteration, where they did receive the combination of lapatinib and trastuzumab based on the data generated in breast cancer. But I mean, this was the treatment algorithm we used, and we kept stick to this treatment algorithm all along the trial. So we were very stringent, and this was part of the treatment algorithm as well in terms of variants of interest for mutations. So we are looking for validated hotspot mutations, and you can see that we had decided on thresholds for the frequency of the mutation or the coverage, depending on whether these were SNVs or indels. But here again, you might tell me, or I would probably have not used like 4%, but rather 6%, or not 30x, but probably 50x. And maybe, but nobody knows what is the true answer to that, right? And this was the same for gene copy number alteration. So we are looking for numbers exceeding 6 for diploid tumors and 7 for tetraploid tumors. But also we didn't want to have large amplicons because we wanted to be sure that our genes of interest was the one that was amplified or deleted. So if the amplicon size exceeded 10 megabases, then it was not good for us. Below 1 megabases, the probability that our gene of interest was amplified or deleted was quite high, and then this was fine for us. And between 1 and 10, we wanted to have a confirmation at the protein level. So here again, this was part of our treatment algorithm that was disclosed, and you might have done differently, right? So we included 740 patients. Most of them had a tumor biopsy, and around 70% of patients had a tumor molecular profiling. 30% of patients did not, because the cellularity was too low, so we wanted more than 50% of cancer cells at the beginning, and this was amended to 30% of tumor cells. And then 40% of patients had a druggable molecular alteration, which is 40% of the patients, and 20% of the whole population, right, was randomized. And some were not, because their condition did deteriorate and could not be randomized. So we had decided to include patients with all types of cancer, and we didn't want to have like 60% of patients with the same tumor type, such as breast cancer. So we stopped the breast cancer cohort very early, and we wanted to have like an heterogeneity in terms of tumor types, because this was the principle of the SHIVA-01 trial. The idea was really to compare in routine what would bring tumor molecular profiling and match therapy as compared to what we do in the clinic. So here are the patient's characteristic. What is important here is the prior the number of prior lines of treatment, which was 3 at the time of the biopsy, but patients were allowed to have another line of chemotherapy at that time, not targeted therapy or hormone therapy, but chemotherapy. So it was more like 4 lines of treatment at the time they were randomized into the trial. Second important point is what molecular alteration we did find. And you can see that the most frequent molecular alterations involve the PI3K/KT-mTOR pathway, which we all know is a canonical pathway in cancer, but that is a reality. And many patients also had a molecular alteration in the hormone receptors. And at the end, very few patients had a molecular alteration under what we call the RAF/MEK pathway, where we have all validated oncogenes such as EGFR, HER2, PGGFR alpha, etc. So this is the result. Actually, we didn't find any difference between the 2 arms in terms of PFS. If we look at subgroup analysis, and we did stratify the trial based on the hormone receptor, PI3K/KT-mTOR pathway, and RAF/MEK, optimistic people in the room would say, oh, the curves separate a little bit. Yeah, I mean, that's true, but it's not significant, and it is a subgroup analysis, so we cannot draw robust conclusion based on that. So we're now working on that in our SHIVA-02 trial, but this needs to be confirmed in another trial. In that group, there were many molecular alterations involving EGFR, HER2, KIT, etc. So what are the challenges of implementing precision medicine? And the biggest challenge is the treatment algorithm. And the treatment algorithm is how we allocate treatment to patients depending on the molecular alterations we find. And the treatment algorithm has several features. First, what is the technology that is being used to identify molecular alterations? And we know that depending on the technology, we might find different results. So obviously, it's not the case for the hotspot mutations in our genes of interest that are very few. But I mean, this is a study where the same sample was analyzed with 3 different machines, and you can see that not all alterations are common between the 3. The second is the thresholds that have been used. So I did tell you the threshold we used for frequency for mutation or coverage, minimum coverage, or number of genes for amplifications, etc. Now, if you change the thresholds, you might change the result as well. And here is a study where a single sample was analyzed with 5 different pipelines with the same machine. And you can see that depending on the pipelines, only 57% of molecular alterations were common. So it means that depending on the thresholds, you might change the results. So again, the technique you use must be disclosed. The thresholds that are being used must be disclosed as well. The third is how is the matching between molecular alteration and drugs. And that is a very tricky part. I didn't put here the paper by David Heman published in Nature like last week with neratinib being given to patients with any kind of HER2 or HER3 mutations. And that's not easy. Not 100% of patients do respond. Most of patients do not respond to neratinib when they have only a HER3 mutation. And now, when they have a HER2 mutation, only part of the patients do respond, and actually a minority. And it depends also on the tumor type. So it's not like it's something that is easy. The main bias is that our treatment algorithm in that neratinib study or in Shiva or in many trials that they are unidimensional. So we have one molecular alteration for one drug, but we know it's much more complicated than that. We know that patients who have a PI3K mutation, if they also have a KRAS mutation, they won't respond to a PI3K inhibitor. And that's only a bidimensional treatment algorithm. But probably we should look at the whole picture and look at all the coexisting molecular alteration to try to guide treatment. Also, I don't know how to do that, right? If you know it, you're good. The last challenge is prioritization of molecular alterations. If there are several molecular alterations, how do you know you have to target EGFR first or another one? So obviously you would like to combine drugs, but medical oncologists in the audience know how difficult it is because of toxicity, right? Or you might lower the doses, but then the drugs are no longer biologically active. So that's an issue. So in conclusions, I would say precision medicine has successfully started with the assessment of single molecular alterations in specific tumor types, actually. It's Herceptin in HER2 positive breast cancer patient. It's erlotinib in EGFR mutated lung cancer, crizotinib, etc., etc., etc. For this kind of drugs, we do not need high throughput technologies, right? Now, high throughput technologies have been shown to help prognose, and that's the case for early breast cancer with the gene signatures such as MammaPrint and Oncotype DX, which is already marketed for a long time now. And high throughput technologies might have a predictive value, but this needs still to be demonstrated. So today nobody can tell whether a patient who will get molecular profiling will live longer than patients who won't get molecular profiling during their disease course. And we really need to demonstrate that. Now, it is important to do it for including patients in clinical trials, right? But not for giving off-label drugs. So at the Institute Curie, we do molecularly profile all of our patients with recurrent cancer, but in order to guide them to specific clinical trials, not to give them off-label drugs. So that's my take-home message, and I thank you for your attention. I'm happy to take any question. Thank you.